So welcome to the next in the monthly series of the UK International Coronavirus Network seminars. It's great to see so many people here. Welcome. As I'm sure the Zoom voice has just reminded you, this is going to be recorded and will be available on YouTube later. I'm really pleased that we've got two excellent speakers this morning. The second speaker, Dr. Hazel Stewart, is going to be talking about tethering antagonisms by SARS-CoV-2 or 3A and spike protein and how that enhances virus release. But the first talk is from Dr. Craig Duffy, who's going to be talking about the application of Nanopore and Illumina sequencing in infection biology. Craig's a population biogeneticist and bioinformatician working at Liverpool. He's got a background in African trypanosomes and malaria. More recently, he's been working on Mpox and DRC. And I think we should be promised a really excellent, interesting talk from Craig around the utility of sequencing. So without further ado, let's get on with that. Craig, I think that you've already tested your slide sharing and everything, so hopefully we can just pass over to you and let you get on with it because that's what people are here to listen to. Fantastic. Good. Craig, over to you. Thank you very much. And then we'll take questions after Craig, and we'll, if you can use the raise hand function for thinking about questions, you can put them in the chat as well if you want to put them while they're fresh in your mind during the talk. But I think Craig will be expecting to deal with questions at the end of his talk, not halfway through. There are one or two of you who are not muted. Please, can you mute yourselves, or Jill, maybe you can mute people so to make sure we have minimal background interference for Craig. Thank you very much. Craig, over to you.
Well, good morning, everyone. So, as I say, I'm Craig Duffy. I'm going to be talking about sequencing and infection biology with a focus on Oxford Nanopore and Illumina platforms. This is going to be a bit different from most of the presentations that the network has during these talks because I'm not really talking about the work that I do or the results. I'm more focused on the technology, the application of it, and what I really want to do is get you to think about what sequencing approaches are out there and how you can use them for COVID sequencing and infection biology in general. So, as already been mentioned, I am a molecular geneticist, population genetics and bioinformatics. I spend most of my time these days as a data analyst, but I don't come from that sort of computing background. I come from a biochemistry, molecular genetics background. So I've got that view of, I know what the challenges in the lab are, and then the challenges when it comes to the analysis, which is quite a useful view when you're working with people that don't necessarily have an informatics background or vice versa, don't have a molecular background. I find myself a lot of the time supporting other projects within the lab saying, well, this is the limits of this. This is how we get around it. I started as a parasitologist working on the African trypanosomes, then moved to malaria with my first postdoc, working in West Africa, looking at how sequencing could tell us about the selection that was on the genomes of West African malaria. When I moved to Liverpool, I came back to the trypanosomes, looking at the complex process of the recombination of the antigens that they use to hide from the immune system. And now, as part of the Hiscox group, I'm one of two bioinformaticians in the group looking at viral infections, supporting other members of the team, and using bioinformatic approaches to understand the evolution of the viruses, COVID in particular, over the last few years, but also host responses to viral infections. And if anyone really was doubting my nerd credential, yes, I play D&D as well. So as I say, I'm not really going to talk about results here. I'm going to be talking about sequencing platforms. And I want you to be able to come away from this talk thinking about, well, I know a bit more about these platforms now. I know which one I might want to use for my um, experiments going forward. And there's really three options when it comes to sequencing. We've got Illumina, tried and trusted stalwart, that has its limitations in terms of its very short read length. So you're generally only getting fragments of 150 base pairs. At the opposite end, we've got PacBio and Oxford Nanopore, which are the current third generation sequencing technologies that are designed around longer reads. I'm not really going to talk about PacBio today. It's not something we use widely within the group, and it's a really expensive platform. The machines cost upwards of £1 million, so they're really only seen in sequencing centers. Whereas in the Hiscox group, we use a mix of Illumina and Oxford Nanopore. So I'm going to focus on those. And Oxford Nanopore is one of the new machines that is coming out over the last 10 years. It's focused on medium to ultra long reads. So we're talking 200 base pairs up to in excess of a megabase. And it contrasts to Illumina in a lot of ways because Illumina, we've got ultra high accuracy, tens to hundreds of millions of reads. Nanopore, we've got a lower accuracy, low to medium throughput for a few million reads. But we've also got advantages of Nanopore. It's got the shortest runtime. It's got real-time data acquisition, which we used extensively during the pandemic to quickly generate results. And it's a very different technology. It's electrical sequencing that's still developing as opposed to the mature sequencing by synthesis technology of Illumina. It's essentially the same process of Sanger sequencing, just scaled up to 1 million times. So before I really go any further, if you come away from anything from this talk, and I hope most of you come away going, oh, I already knew that, he's talking mostly sense. But what I really want you to come away with this is talk to a bioinformation at the start of your project when you're planning it. Because time and time again over my career, I've been approached by people that have generated all this data and they go, how do I analyze it? What do I do with it? Why isn't this working? And this ranges from I've had PIs that I don't work with go, I've got this pile of data, but nobody to work with it. Third year students that have spent the last 3 years generating mutants or analyzing an infection, and now they don't know what to do. Or people that have done their experiment and they go, I didn't add any controls. How do I do that? So I really want you to come away from this thinking, this is a complex process. I need to talk to someone beforehand. Could be a bioinformatician within your department or the sequencing group that you're working with. If you're working with a sequencing center in particular, they will have people that can advise you that say, you're aiming to sequence COVID in 1,000 people. This is your budget. This is the best technology. This is how you analyze it. So get in there early and think about it before it's too late and you're scrambling to catch up and generate a result that makes sense. So I want to spend a little bit of time just going over the pros and cons of the two platforms. And if you're familiar with sequencing, a lot of this will be quite obvious. But I know we've got a mixed background of people here, so I wanted to spend some time on it. So Oxford Nanopore, which we use regularly, is a really versatile mixed system suitable for short sequencing up to the ultra long reads. It's also got a really advantageous method over Illumina that you can directly identify base modifications. So if you're interested in methylation or whether a new antiviral drug works by tweaking the genome with a modified base, for example, you can identify that directly with this sequencing platform. It's also cost effective at the small scale. So the smallest sequencer, the MinION, which is normally where I pull this out of my pocket, is no no bigger than a USB stick. It costs under £1,000 for the sequencer. It plugs into a laptop. It's quick, it's effective, and it can fit easily within a standard lab setup for on-demand sequencing. So we use it a lot for sequencing viral stocks that we're growing, checking that they haven't mutated. We can have that done from generating the RNA through to getting results, potentially in under a day, normally in 2 days. So it's a quick, accessible technology. The cons of it, though, as I say, it's got quite a high level of variability in terms of quality and quantity of data. So if you're doing a standardized assay where you need X amount of data for every time, you're going to find it varies quite a bit. Of all the sequencing platforms, it's also got the highest per base error rate, which I'll come on to in a bit. But this is obviously going to impact your choice of what am I using it for and how much data do I need to generate. Chimeric reads are unfortunately common. So this is where you get two pieces of DNA that have no relation to one another, joined together and get sequenced as one piece of DNA. There are difficulties dealing with repeats, and there are limited multiplexing options. At the moment, you can sequence up to 96 samples in a single run. Illumina, you can go substantially higher with that. So if you're looking at a population-wide survey, you've got thousands of samples, only doing 96 at a time is going to be extremely challenging. It's also a still developing technology. So the kits, the chemistry it uses are not in a fixed state, which means it's got limited validation for clinical use. This is starting to come on board, but for clinical studies where we're looking at potentially mutations or variations in the host in terms of how they respond to a viral infection or the use of drugs, it's going to be harder to validate and present that in a way that both funding and regulatory bodies are going to be happy with. So those are the main cons of these two, of the Nanopore platform. On the other hand, we have Illumina, which is a tried and true established technology. It's got the lowest cost per base, but only once you scale up. So if you're sequencing one sample because you're using it in the lab and you want to check what the sequence is, it's not really cost effective. If you've got the 9,000 samples on a single run that's now possible, it's extremely effective. Your cost per sample comes down to under £100. It's also highly accurate. We're talking about in excess of 99.9% accuracy. Sorry. Established analytical pipelines and clinical validation, and extremely high read depth is possible in a cost-effective manner. So just to give you a quick comparison in terms of depth and cost, for an experiment we're currently looking at variation of host transcripts for 100 samples, 65 million reads per sample. With Illumina, we're looking at about £15,000 to get that sequenced at a sequencing center. Using Oxford Nanopore to get an equivalent read depth for the same samples, we're talking £40,000 just for the flow cell reagents. This doesn't include handling, doesn't include any depletion steps we have to do, all of which were in that £15,000 for Illumina. So you can see for large surveys where you need high read depth, Illumina is often a much better platform. There's a lot of cons though. The biggest one is the short read depth. We're only getting about 150 base pairs on average, which makes it extremely limited for genome assembly or when we're talking about complex haplotypes especially for MHC regions. Illumina is very limited, whereas Nanopore, with the longer reads, you can sequence an entire gene in a single read, get the haplotype, and compare it to other haplotypes. There's also a long turnaround. If you're running the machine directly, you're talking at least a week. Obviously, when you do a sequencing center, that then scales up because it goes into a queue. You might be waiting 3 months to get your data back, compared to 2 days for Nanopore. The machines cost a lot more, there's a large initial investment, and there's no direct ability to sequence modifications if you're interested in that. So really, when it comes to choosing your platform, I want to make sure that you're thinking about 3 things. The cost. What's your budget? We've all got limited budgets for a lot of this work, but that's a really important factor. How quickly do you need the results? As I say, if you're using a sequencing center, they may be cost effective, but you might be waiting 3-6 months to get the results. But the really big one is, what are you sequencing this for? Why are you doing this sequencing? And I just want to talk about the 4 main aims and sequencing approaches that we use in the Hiscox group as part of our work, and talk about the limits and when you should think about these. So these are whole genome sequencing, metagenomics, transcriptomics, and targeted sequencing. So whole genome sequencing is used primarily for 2 factors. First is assembling the genome of a new species, a new parasite, a new virus. And for assembly of genomes, longer reads are key. So this is where Nanopore really shines, and it's far better than Illumina. We typically, in the group, only assemble viral genomes, so talking 30 kb on average for what we're looking at, but Nanopore, you can assemble human scale genomes far easier than Illumina ever could. The other approach for whole genome sequencing is diversity. SNP calling, trying to identify variants that might be associated with long COVID, that might be associated with response to antivirals. For this, Illumina remains the gold standard, and it's highly accurate even at lower read depths. And again, there are established and validated pipelines, but one of the things I'm not really going to talk about today is to think about how computationally intensive and the data servers that you will have to have for a lot of this bioinformatics. Targeted or amplicon sequencing is something that we used extensively during the pandemic with the Arctic pipeline that was widely adopted for COVID sequencing using Oxford Nanopore. And essentially, this is using overlapping primers to piece together the jigsaw of the genome and identify variation. Nanopore was really useful for this because it allowed us to amplify our areas that we were interested in and rapidly get results and identify the strain of COVID that we were dealing with and feed that into the national network. But one of the things that we've seen is that optimization of the primers can vary wildly, and as a result, sequencing depth can vary wildly. And one of the things we're trying to do at the moment is further optimize our approach to sequencing. So the figure you can see here is just the COVID genome, and you can see the depth of coverage spikes in certain regions. And this is simply down to optimization of the primer protocols. So we're trying to see if we can reduce that, level out the coverage, optimize the data we're getting so that we're not wasting resources, so that we can do sequencing in a more cost-effective manner. Related to this, we have metagenomics, what's in my sample sequence approach. So here, this is looking at a bulk sequencing, trying to work out what is there when you don't know what's there. COVID obviously took the world by surprise. We didn't know it was present in the host organisms, so we weren't looking for it and expecting it to jump into people. So a metagenomics approach, we're looking at how this can be used to identify viruses or bacteria in host animals, in bats. We're working as well with MERS to look at how it's present in camel populations in the Middle East. And using this approach of sequence everything, quick turnaround with Nanopore, we can ideally go into a market or a herd of animals, take samples, get our results quickly, and say, these animals have this infection, we need to deal with it. Nanopore is great for that, but the error profile is causing challenges, leads to misassignments of what species are present. And I won't go into this figure in depth, but this is just an alpaca sample where we were looking at what bacteria and viruses were present. And we actually ended up picking up viruses we'd worked with in the lab that we knew weren't in these samples because of cross-contamination, because of how sensitive these techniques are. Finally, for the last few minutes, I want to talk about transcriptomics, which is the area that I'm mostly working on. And here we're using transcriptomics to look at how individuals respond to disease by changing the expression of their own genes. So we're using RNA-seq, sequencing mRNA to look at those changes between an infected and uninfected person. And here the factor that we're finding is that the most important factors are the depth of sequencing and the number of controls. So for Illumina, we've got well-established pipelines and recommendations. We are recommended a minimum of 30 million reads per sample for humans to do bulk analysis of gene expression. Even more if we're looking at rare transcripts. We've been keen to move to Nanopore technology, where it's suggested we need about a third of that. So we're talking roughly 15 million reads per sample. And from our work in the lab, we've found that this is roughly equivalent to an entire MinION flow cell, or about a fifth of the larger Promethean flow cells. But we're lucky that there's been a couple of bits of validation. So Massel et al. Used a large data set that they then were able to subset to assess how many reads do we actually need and how variable are our results if we only take 10% of that data, 20%. Because the majority of Nanopore papers are using only a small number of reads. And they found that they needed roughly 14 million reads per sample before you lose the variability in terms of your results. And thankfully, this is actually very close to what we've been estimating. So it's a good cross-validation with another group. But what we found, though, as I go back to those numbers, the cost effectiveness of this is just simply too high. So for Nanopore, we're actually shifting away from Nanopore for looking at our host response, keeping focused on Illumina because we want the depth of coverage, because we're doing statistics to compare how different is the expression of this gene to this gene in the other person. And for that, you need bigger numbers are better because bigger numbers give you bigger p-values. But obviously, depth of coverage is just one factor. We need to consider the number of controls and the strength of the effect. And here, this is something I've dealt with people coming to us at the end of an experiment saying, I've got 2 controls, is that enough? And the answer is no, it's not enough. You also need to think about how different your samples are going to be. So on the left, I've got what's almost an optimal PCA plot comparing different samples with a transcriptomic approach. And you can see in the top left, the blue separate out nicely. The red separate out partially, and the purple and green are very similar to one another. So here they've run an experiment where they've got a large amount of variation and you can easily separate the samples. More common though is the example on the right, where on a bulk level, it's basically impossible to separate infected from uninfected people. There wasn't enough power in the experiment to do the analysis they wanted because they hadn't really considered, even with a large number of controls, how big is the effect? What do I need to consider? So I really want people to come away from this thinking about, I need to plan this at the start and consider this at the start. So just to summarize, and I hope most of you have sat here thinking, I know all this already, I'm doing this already. But please talk to a bioinformatician and the sequencer when you start planning your project. Make sure you have enough controls and sequence to a depth that's appropriate for what you are doing. Nanopore is great for a quick turnaround and day-to-day use if you're sequencing samples that you're growing viral stocks. And in the Hiscox group, we use it a lot. We're happy to advise on, these are primers we use. These are the protocols we use. If you're going for big projects where accuracy is vital, stick with Illumina. It's cost effective, it's accurate. The thing I haven't really touched on is the informatic analysis. It's complicated. It's not something a PhD student can learn in the last 6 months of their project. Think about it early. Think about whether you've got server capacity. Think about the data storage if you're generating hundreds of gigabytes of data. Again, think about this at the start. So I just want to thank everyone in the Hiscox group, especially Julian, who took me on board and brought me over from the dark side of parasitology to viral genetics. All the postdocs, technicians, PhDs, and administrators in the group, past and present, and obviously all our funders that make our work possible. And thank you for listening.
Craig, thanks. That was a fascinating run through different approaches. You may see that there's a question from Kevin in the chat. Kevin Bewley, does Nanopore still have issues recognizing or mapping deletions and insertions? And is that the sequencing tech or the mapping software? I don't know if you feel you've answered that now, but I wonder whether you could just quickly go over that, or I wonder if Kevin wants to add anything further to that question.
So a quick, yes, not as bad as it used to be, but it does still have issues with insertion deletions, and it's a technology side of it rather than mapping. So the way the nanopore sequencing works is that as it passes through the pore, you get a change in electrical current. But it's not measuring it on a per base measure. It's measuring approximately 5 bases at a time. And sometimes it jumps a base. So that's where your insertion deletions. And it also comes in if it's a repeated base. It sometimes can't always tell that the DNA has moved. So it will say, oh, there's an A at the end of here. I'm moving along a little. Oh, there's another A. It's the same A, when it might be an extra A. The issue is mostly with small insertion deletions, with 2 to 3 bases, maybe. For the larger insertions where you're talking about a gene duplication or a gene loss, it's great because you get it in context. You can sequence a whole gene and then see there's an entire another copy of the gene on the same piece of DNA, which just isn't possible with Illumina.
Thanks for that, Craig. I guess the context for my question was during some of the variants emerging during the COVID, sort of the evolution of the pandemic, when something came along that had a big gene deletion, Nanopore was often our first line of sequencing because of the speed of it, because you can get it done in a couple of days. And then we weren't sure, well, is there something wrong with the primers now, or is it a deletion? Is it a true, like Omicron had a huge deletion in it. But yeah, just curious whether that's something that will eventually be cured if it's software or if it's just a limitation of the technology itself, really.
Yeah, I mean, it will. It's getting better. So Nanopore is very much a developing technology. Um, the leaps they made during the pandemic, thanks to the amount of data and people using it, was massive.
Mm.
So I think they will get there eventually. I just don't know when. Um, my advice at the moment would be if you're worried about that, over sequence
Yeah.
because it tends to be quite a random process. Um, so you might, if you've only got 5 reads, you might not be sure. If you've got 5,000, you can say this is the majority.
Thanks, Craig. Great talk, by the way.
So,
Thank you.
so we're spilling over a little bit past the half hour. Louise, you half put a hand up before Andrew got his up. Is it a
Hi.
quick question?
No, it was more very briefly about library prep. If you want to look for DNA, RNA viruses and bacteria all at the same time, would you advise doing two different preps where you know you do an RNA prep and a DNA, or can you approach it in all one kind of prep?
At the moment, I would recommend two separate preps because you need to separate out, obviously, your RNA to then reverse transcribe. So you're already separate. So I would, at the moment, recommend keeping them separate.
Okay, thanks.
Thanks.
Sorry, a bit tangential, but
No, this stuff's really important, Louise. Thank you.
yeah,
It's a good
yeah.
point. Andrew, you've disappeared and taken your hand down. I wonder whether you could ask Craig a question in the chat. There are some answers and questions and comments in the chat, which I'd refer you to just as I'm start to introduce our next speaker. But before I do that, can we all thank Craig very much in a time-honored Zoom way for a really interesting, excellent talk before we move on to talk two, please. But no, that's great, Craig. Thank you very much.